All right, thank you, folks. I feel very connected to you, so hopefully you can hear me double-sided. I appreciate the introduction of President Albrecht. It has been a wonderful opportunity to be at Utah State, not only as a researcher, but also an administrator under the president's leadership. I appreciate you folks all coming out on a fall morning and a Friday to boot. So hopefully I can get you started with some cool research that'll send you off into whatever your day holds. Let's see. We'll try some buttons. Oh. All right, I'm gonna have to do some things here. All right, I need some technical assistance. There we go. All right, well, as you heard, my talk is on the sheep genomics project that I've been doing for about the last 20 years since at Utah State University. I thought, though, for those of you that don't know me, I'd give you a brief background on myself and how I ended up doing this area of research. I grew up on a ranch in eastern Montana. We had sheep until I was in 4th grade, and then we sold those and bought beef cattle. When I was in high school, we didn't have really the best high school counseling. And so as a student with good grades, I was offered 2 options: to be a medical doctor or a veterinarian. And since I loved animals, I thought a veterinarian was what I wanted to be. So I went to Montana State in animal science and discovered, though, that genetics combined the 2 things that I enjoyed most of all, and that was biology and math. So I continued in animal genetics at Oregon State, spent a brief time in Nebraska, and then came to Utah State in 1990. And when I arrived, the beef cattle were actually all assigned to nutrition projects, and so they told me that I should consider working in sheep. And while it was a little bit of serendipity, I'm very, very glad that that's the direction that I ended up taking. I'd like to give just a little background on sheep production in the world. Right now, there's about 655 million sheep in the world. It isn't certainly not the largest livestock species. In fact, it's 4th in the world. With chickens, there are about 37 billion, swine 3.5 billion, cattle 1.3 billion. So you can see that sheep is a distant 4th. Unfortunately, in the US, the numbers of sheep continue to decline. And also the age of the producers working with sheep seems to be declining. There's only about 84,000 sheep operations left. Part of this is, some concern about the prices of lamb and wool combined with increasing costs of production. But I also think there is a decline in actually people eating lamb. So next time, do me a favor. When you see it on the menu, please order it. Now, sheep has turned out to be interesting from the amount of genetic variation that exists. And the reason is, one of the cool things about sheep is you can actually get 3 products out of an animal: meat, milk, and also wool. Now, very few breeds have actually been developed to produce excellence in all 3 things. Rather, you have certain breeds that are emphasizing the meat, certain on the milk, and certain of the wools. So you have lots and lots of genetic variation broken up into breeds. Also, sheep are a very small unit and can be raised on fairly harsh environments on submarginal lands. And so you'll find it all the way from the peaks of Iceland to the Caribbean lands. And so it's also turned out to be quite useful for third world countries and small, very, very small operations. Now, when I introduce myself as working with sheep genomics, for some reason, people always ask me if I do cloning. Now, cloning is where you take an animal's DNA and propagate it into another animal. So you end up with a series of identical twins, but that may be born at different points of time. Actually, it turns out that I think that's pretty boring. And the reason is because they're all identical. There's no variation there except from the environment that they are found in. Instead, I'm interested in the differences that animals or humans have, the genetic variation that exists. And this genetic variation is what makes a zebra and a sheep, and even more interesting, what makes individual animals within that species different. For instance, if you look closely at those zebras, they actually have different striping patterns, which is caused by their genetics. I'm going to spend just a little bit of time kind of giving a background on where this genetic variation lies within the DNA molecule. Now, the DNA that is found in every one of our cells is composed of these molecules. We refer to them as bases, and we abbreviate very long technical names into 4 letters: G, C's, A's, and T's. In each cell of your body, there's 3 billion of these A's, G's, C's, and T's, and every one of your cells has very, very similar composition. And it's this order of that G, C's, T's, and A's that defines you as what you are, differentiates you from a sheep or a honeybee or whatever. Now, as time goes on, your cells of your body are constantly being replenished. And every time a new cell is made, the DNA is also reproduced. And so your cells differ in their DNA content than your neighbors there at the table. But as this is done, this replication of DNA occurs, sometimes that order, that very precise order of those bases changes. And those are mutations. Mutations are often neutral. They aren't even noticed, but sometimes they really do affect a trait or cause a disease or make a real difference in that DNA molecule. Mutations occur at about 175 bases per generation, so that as generations move, more and more mutations occur, and that's what causes the divergence in breeds or organisms. And then finally, the DNA codes for proteins. It's actually the proteins that are making you a human and a sheep a sheep. They can be muscle proteins, enzymes, hormones, regulators, whatever it is. But all of those proteins trace back to the DNA. So if you know the orders of the C, G's, A's, and T's, you can predict the proteins and you can predict the traits. Now, I have on the tables, there's some racks there. We were going to do a little experiment, but I thought that might be too early on a Friday morning. So instead, what I've brought are tubes of DNA. So I encourage you to pick up those tubes. Maybe don't drink them, but pick those tubes up and just roll it in your hand a little bit, and you'll actually be able to see the DNA. It looks like white, fine dental floss. It's quite easy to obtain. Since all of the cells of your body have it, any of your cells could be broken open and release the DNA. We find blood the easiest thing to get DNA out of. And so when we're doing a population of sheep, we go in, collect 10 cc's of blood, and we would be able to extract about that much DNA from it. But obviously, it's not a good process to have that tangled mess of DNA in each one of your cells. So instead, it's very carefully packaged in things that are called chromosomes, where those DNA strands are spooled around protein and held in a firm structure. Now, the chromosomes are very, very precise. I'm showing the chromosomes for sheep. They're not very imaginative. They're just numbered 1 through 26, along with the X and the Y gene that control whether the animal is a male or female. But across sheep, the genes, the pieces of DNA that are found on chromosome 1 are found on chromosome 1 of all sheep. And so by knowing what order of these genes that produce which proteins are on chromosome 1, we can start diving down into what the genetic variation is. The last thing I want to mention, sort of this primer of genetics, is that in every one of your cells, you actually have 2 copies of every chromosome. One set came from your mother, and one set came from your father. And it's that combination of those 2 sets of DNA that make you what you are. And then in the sperm and the egg, it's only 1 set of chromosomes, sort of randomly distributed, and then creating that next generation. But again, the DNA mutates over time so that while maybe all the same genes are found on chromosome 1, what that DNA sequence is on chromosome 1 differs. And it's that I am most interested in looking at. So just to go through this a little bit more, I've got a couple of examples of things that are controlled by DNA mutations. The first one is an example of a single gene trait, one that only 1 gene found, it happens to be found on chromosome 10 in sheep, controls whether the animal has horns or not. And it's just a single change in that DNA sequence that causes that. Now, this one is easy to look at, easy to work with, because it's a single gene. The other thing about it is that environment doesn't affect it. No matter how you feed the animal, no matter where it's raised, no matter what year it's born, it's going to have horns if the DNA says horns, and it's going to be polled if the DNA says polled. But a lot of the genes that we're most interested in are actually in animal production, are actually those that are called quantitative traits. These are traits that are controlled by multiple genes and also have an environmental effect. For instance, when you look at wool, there's the length of the wool, the size of the wool, the texture of the wool, and it gets more complicated when you look at things like meat. Where is that meat produced? How much fat does it have? What are the flavors in that meat? And then finally, lambs. How many lambs an animal produces or raises? And again, these traits are very important to us from a production standpoint, but are more complex because of multiple genes and the environment. So when we look at sheep genomics, this is a branch of genetics that is looking at the DNA to discover what the genes are that are controlling those traits. And as I mentioned, I work in the agricultural sheep genomics because I'm interested in the DNA that's affecting things that like meat or fiber or milk. There are other people, though, that are working in the life sciences using sheep genomics techniques. And they're interested in this vast genetic diversity that exists across all those breeds of sheep that are raised from Iceland to the Caribbean. And then we also use sheep for models of biomedical interest. There are many diseases in humans that can be modeled in sheep: kidney disease, asthma. There's actually studies in sheep for fetal alcohol syndrome. And also they have some mutations, some disorders that causes diseases similar to humans. those that affect the eye or the bone or the muscle. So that by studying sheep, you can actually predict what's happening in the human and lead to better treatment of diseases. Now, to work through this, I'm actually going to show you some results from our lab on a trait called Spiderland syndrome. It was actually done a number of years, but I think it's got a good story as we move out, and how a production trait, something that was deleterious in sheep, actually has translated into some research in humans. So as you can see, the little lamb there has a bone disorder. It has extremely elongated legs. It also has curvature of those legs. It has curvature of the spine. Its sternum is malformed. This is called a recessive disorder because it actually takes the mutation that's passed from its mother and the mutation passed to its father to come together before you actually see the lamb with the defect. This is its mother, looks normal. It's a carrier for the disorder. It has one copy of normal, one copy of spider lamb, and so the normal is masking over its effect, and it's not till it's produced a lamb, a defective lamb, that you know it has the mutation. But in the meantime, it's producing other lambs that look normal. So this was actually traced to a ram that was used quite heavily in the Suffolk sheep, the black-faced sheep shown there. It was born in the 1970s. And in the 1980s, it was realized that it was a genetics disorder. And then when I moved to Utah State, I was asked to find the DNA that causes this, because the hope was to have a genetic marker test that could be done on lambs at birth to discover whether they were carriers of the defect. And if you knew that about an animal, you may not keep it in your flock. So we collected samples from hundreds of these spider lambs and their parents, and we asked what part of the DNA differs between those that have the defect and those that don't. And it actually took us about 3 years to systematically go down chromosome 1 and chromosome 2, chromosome 3, and compare the DNA sequences across those groups of animals. Eventually, we found a difference that predicted whether the animal would be a spider or a carrier or completely normal. And it was found on the bottom of chromosome 6. Now, at this time, very little was known about the sheep genome, the DNA of the sheep. And so we had to move to a different species and see if there was something there that could help us untangle what the cause was. Well, on human chromosome 4, the genes and the DNA is similar to what's found on sheep chromosome 6. So we were looking for something that in that same location, that same position, would be similar to what we saw in spider lambs and therefore tip us off on what the defective gene was. It turned out that in that same region on human chromosome 4, there's a defect, human chondrodysplasia, or dwarfism. It's actually a mutation in the FGFR3 gene, the DNA sequence that produces this protein. This protein normally is activated through a process when bone length has reached its maximum. And so it's involved with turning off bone growth. In the human chondrodysplasia, there's a mutation in that DNA that makes that protein appear and be activated, shutting off bone growth too early. And in this case, an embryo that was lost before it reached term. Now, in our minds, it was like, well, it's involved with bone growth, but it's actually the opposite effect. We don't have bone growth in spider lamb that's too short. It's actually too long. But in at that point in time, researchers who were studying human chondrodysplasia had actually created what they called gene knockouts in mice. And they had gone into the DNA of mice and knocked out, actually snipped out, part of the gene for FGFR3. And what I'm showing you here are some of the normal mice compared to those mice where that gene was knocked out. And you can start seeing similarities with what we had in our spiders. The curvature of the spine, the oddly shaped sternum, and then actually longer bones in their legs. So because this was very similar to what we saw in spider lambs, we decided to pursue it further. And we actually sequenced then the DNA, determined the orders of the A's, G's, C's, and T's for the FGFR3 gene. It was about 150,000 bases that we actually determined. And when we lined it up to the DNA in humans for FGFR3 and the DNA in mice, we noticed that there were differences that occurred for the sheep, but there was one base that changed when we compared the spider DNA to the human, the mouse, and the normal sheep. And all it was in this 150,000 bases, instead of the DNA reading GTG, it reads GAG. And every spider we sequenced had it, and every carrier that we sequenced also had one copy of that defect. Now, I mentioned before that the DNA codes for proteins, and proteins are made up of amino acids. So a triplet of DNA tells an amino acid to be attached to a protein chain. In this case, that single GAG mutation changed the amino acid in the FGFR3 from a valine to a glutamic acid. And that's all it takes to create that mutation in that protein that causes that trait in those spider lambs. So one of the things that we wanted to determine was how prevalent this mutation was. So we went to a national ram sale and asked if producers that were showing suffix, where this original mutation was found, would let us take a blood sample from their nationally ranked ram. We collected 114 samples from 23 breeders, and very, very surprisingly, almost a quarter of those animals actually were carriers for that defect. They looked normal, but they were causing that defect, or they were carriers. And no doubt somewhere in that pedigree, those producers had found spiders. So then we said, well, let's see what's happening in the rest of the purebred black-faced sheep. These would be Hampshires, South Downs, Shropshires, and Oxford, again, at this national ram sale. So in total, we got 336 samples for 61 different breeders, and on average, 29% of those animals were actually carriers for that spider lamb defect. Now, one of the things that this did was send shockwaves through the industry, because when you are showing a purebred Shropshire, you shouldn't have the genes for something that's actually a mutation in suffix. So a lot of people did get caught having done some crossbreeding of Suffolk with their breed back in their previous pedigree. But the other thing that was so stunning was how high the frequency was. And so we talked to people and we said, yeah, every time you tell us you've got a carrier, it's one of our better animals. And we said, well, what is it that you're considering to be better? And it was frame size. So we did a very, very simple experiment. We actually took spiders, carriers, and the homozygous, the copies of only normals at birth, and measured their bone length on x-rays until they were about 3 months of age. And it turned out that the carrier animals that looked normal actually had bone growth about 1 to 2 centimeters longer than the complete normal. So anytime a producer was going out there selecting for an animal that had larger frame, that was bigger, they were selecting for carriers. Now, we did set up a genetic marker test. We still get royalties through USU for this genetic marker. And the thing is that producers are actually still breeding for these carrier animals because they want that larger size. But they're setting up the genetic marker test so they don't breed a carrier times a carrier and end up with the defect. Okay, so I'm now going to move into some other areas that this kind of information is being used. And over here, I'm showing you a sequence of a DNA molecule on the top from one animal compared to the DNA sequence on the other. And this new technology is just spitting out what that base is when it's attached to a colored dye. So the top animal actually, because remember every animal has 2 copies of DNA molecules, has both a T and a G at that position, where the second animal only has a T at that position. Well, sequencing that 150,000 bases took us months at that point in time. But the technology is getting much, much quicker. And you can actually generate millions of bases of DNA sequence in a day. But what I'm interested in is the variation, not the sequence itself. What is the variation? So they've come up with a new technology that allows you to detect that variation in DNA through a process called SNPs. And it's in the lab. You synthesize short fragments of DNA. One version of it has one of the base sequence, the other version has another. And they're attached to a small glass slide. Then you take your DNA of the animal that you're testing, that would be shown in red, and you spread it across all of those synthetic pieces of DNA. If it can pair up to it, it attaches. If it's different, it can't, and nothing shows up. So in this thing right here, I'm showing for that particular animal what DNA sequences it has. And this now is placed in a gene chip, is what it's called, where thousands and thousands and thousands of pieces of DNA can be tested for what their variation is. And I've thrown some of those on your table. I think there's a cow SNP chip and there's a human SNP chip. And so at some point, these have actually been used by our service lab up at Utah State, where samples from cattle or samples for human are tested with that chip. And you can now put 50,000 pieces of DNA on one of those chips, and in humans, they're even as high as 1 million pieces of DNA. It takes about a half a day to run multiple animals, and it costs anywhere from $100 to $200 per sample. So that the volume of data that you can get on a single animal is just exploded in the last couple of years. So the next thing I wanted to show you is some results from that type of experiment. From the spider lamb, we were doing 1 DNA sequence at a time. Now we can amass 50,000 pieces of information in a single afternoon, and really the limitation is how to analyze that. So for my example, I've actually shown you a project that it's a mutation in the breed Pyrrondales that causes yellow fat. Now, this would be a normal, this would be a yellow fat animal. You can't tell them looking at it until they're on their pelts removed and they're on the carcass line. But I challenge a consumer who would actually willingly buy a piece of meat that has that color of fat. So it's a genetic mutation. They know it's controlled by a single gene, but they didn't know where it was at. So they took samples of 30 of the normals and 30 of the animals that had yellow fat, and compared them with the results from that DNA chip. So what they're saying is there a difference in which DNA the animals of normal have versus the animal with the yellow fat? And then they plot that for how different those two groups were all across the genome. So each one of those colors is representing the results from a single chromosome. This slide shows from chromosome 10 up through the X chromosome. Now, because you do so many comparisons, 50,000, you can sometimes, by chance, get differences. So instead, you draw a line and say, I'm not paying attention to anything that might be chance. I want something where those two groups are truly different. And it turned out that on chromosome 21, there were regions of that DNA that clearly differentiated between the normal and the yellow fat. And the researchers in New Zealand who are working on this are now identifying the gene, and it's involved, as you would predict, in metabolism and formation of fat cells. Now we're working, this one's kind of, I'm glad everybody's done eating, at least you will be after I explain what this slide is. This is one of the projects we're continuing to work with this kind of information, this 50,000 genetic markers per animal. And that's gastrointestinal parasites, internal parasites. This happens to be the lining of a stomach of a sheep that's very, very infected with internal parasites. And those red strings are actually blood-filled parasites. So I'll quickly get off this slide so you don't have to sit there and look at that. But it's known to be a quantitative trait. Many, many genes also influenced by the environment. And we have taken a cross of animals, Suffolk times Gulf Coast natives. Suffolk's very, very susceptible to parasites. Gulf Coast natives raised in the Gulf Coast have overcome through mutations. They have developed natural immunity to parasites. And now we're looking what the difference is between those two groups using the DNA results from those 50,000 chips. I don't have an answer right at this point. We've got some strong candidates. Most of them are involved in the immune response, and we're tracking down what exactly is going on with this trait. The hope is someday to have a genetic marker test. There are other large genomic studies going on in the world. I've just picked one. I would love if USU had created this flock. This is the Faulkner flock over in Australia. They've generated over 400 lambs produced from 20 sires that have cool and interesting traits in sheep production. And then these lambs were actually characterized for 491 traits. All those animals have been tested, their DNA with the 50,000 SNP chip, and now they're being assayed for looking at what DNA is controlling that. So as these results are coming out, more and more and more data are being found. Just a couple of weeks ago, we actually found what appears to be the gene that controls whether the animal's hoof is striped or not. So, it's really unlimited, the kinds of things that you can discover. Now, what would be the practical implication of doing that? The idea is that you are trying to estimate an animal's genetics so that you can determine whether or not you're going to keep it in your flock or whether or not you're going to use it as a parent of the next generation. Now, currently, the way this is done is that information on that animal is collected based on how it performs and how its relatives perform. And then using that information, an estimated breeding value is determined. Well, with this information, we can actually not only measure the traits of that animal plus its relatives, we now can look at the genetics of that animal. That will be used to estimate a molecular breeding value, and then combined together, you can get a very, very good estimate of its genetic abilities. Now, in the US currently, there's quite a push on developing estimated breeding values for sheep. In fact, I have the father of the sheep breeding values here in the audience, Dave Notter from Virginia Tech. And he and Hudson Glimp, also a guest from out of town, and I are going to be developing a project that we hope USU can be involved in. But in Australia and New Zealand, they have moved forward with determining these molecular breeding values and in starting to use this across the country in evaluating their genetics. And I think it's Hudson, Dave, and my dream that someday we will also be doing that in the US. The last little bit I'd like to talk about is the results from some diversity studies. We've also collected large numbers of samples for 74 breeds that are around the world, as well as wild sheep and outgroup species. And we've evaluated these for different things like when did domestication occur in sheep. And I mentioned that mutation accumulates about 175 per generation. So if you compare groups of animals at where they are now, from wild sheep to domestic, and you calculate back how many mutations have occurred between those two groups, you can actually estimate when they diverged. So doing those kind of studies, researchers have determined that the first domestication of sheep occurred about 11,000 years ago, and these formations of the breeds that we have today occurred about 200 years ago. And looking at archaeological data, as well as just histories of civilization, these are actually very close estimates to what other research has revealed. The other thing that we've looked at is the differences of animals, the genetic diversity. Each one of these circles represents one of those animals that have been done with the 50,000 SNP chip, and then it's positioned with how similar or different it is from all the rest of the animals in the study. Then the second thing that's been done is the breed of the animal is colored differently. So that over there in the black, those are Asian animals. They are very, very different from those in Northern Europe and Southwest Europe. And this kind of data then has been used to look at how breeds have been dispersed across continents. They actually were first domesticated in the southern part of Europe, and then as humans migrated across the European continent, the differences between those breeds became more spread out. So analysis of these are helping us find domestication sites and how breeds have dispersed across the world. One thing you'll notice is the American breeds are similar to the Mideast and the Europe breeds. This again fits with the history of breeds in the US. Now, the final thing I wanted to mention was the holy grail of genomics, which is to determine the DNA sequence for a species. Humans, the DNA sequence was sequenced around the early 1990s. It cost billions of dollars to do that. But every A, G, C, and T and its order were determined on which chromosome. And about 4 years ago, the cattle genome was done. That cost $50 million. Well, we know where we're at in sheep, so we did ours for $1 million. But this continues to be done for all species in the world. There's alligator, there's honeybees, there's platypus, and it's going to be very, very soon where all of these DNA sequences will be done, where you can line them up and compare. So we did ours in a project with the Beijing Genome Institute, who actually planned to sequence and is currently sequencing genomes of 1,000 species. And they had planned just to orientate the pieces of DNA by overlaps, but through some information that we were able to contribute through the consortium, we actually publicly released the genome sequence for sheep in February 2011, and it covers about 91% of the total DNA sequence. And then I could have listed the website, but if you Google this, sheep genome sequence, You could probably come up with this website, and then you can find all of the information for all of the chromosomes, the DNA sequence, physical pieces of DNA, how that sequence was developed, mutations and genes, and so forth and so on. And this is just an amazing resource now that researchers have to begin to uncover the complexity of DNA. So this is really advancing us. But the challenge we have right now, then, is not to reveal the DNA sequence. It's actually to investigate, to dig into all the information that's done. I was mentioning that we did those 400 sheep with the Parasite Project for the 50,000 SNP chip. It actually took us 2 weeks to figure out how to open the data file, because there were 400 animals times 50,000 data points, and we didn't have a computer that was fast enough. So actually what we're seeing with our students is not so much the techniques, because many of these things are actually done at service labs, commercial companies. It's actually how to dig in and compare those DNA sequence differences that we have had. So I think we're going to see a whole new branch, whole new era of genomics where it's simply how to handle the volumes and volumes and volumes. And even in human medicine, it's now possible to sequence a human's genome for $1,000. So that we think that in a very short time, doctors will actually be ordering up a DNA sequence analysis for individuals to identify which kind of genes they have, how they might interact with nutrition, diet, environment, preventative care, so forth and so on. So I think the future of genomic research is going to continue to expand and explore. It's just that we're going to be looking at it in a very different way. So with that, I hope you've enjoyed a little bit about genetics, and I really appreciate the opportunity to present to you today.